for being here. I'm pleased to introduce Marco Keiluweit as our seminar speaker today. Marco is an assistant professor of soil and microbial biogeochemistry at the University of Massachusetts. He did his bachelor's of science degree in geoecology at the University of Tübingen in Germany. And then he moved to the University of Oregon to do his master's and PhD with Dr. Marcus Kleber. He went on to do a postdoc with Dr. Scott Fendorf at Stanford University before starting his position at UMass. His research focuses on response of soil processes to climate change. His group investigates abiotic and biotic mechanisms that couple carbon cycling to other elements. And he works at multiple scales, including using fine scale sensors all the way to the field scale. He also works with fine imaging techniques as well. So we're excited to hear his seminar today, looking at plant-soil microbe interactions in the rhizosphere. Thank you.
Thank you, Hannah. Thanks for inviting me and giving me the chance to present my work today. And thank you all for attending the seminar on a Friday afternoon. I'm sure you're all ready for the weekend. So my talk today will be on sort of a larger suite of projects that I have that broadly focus on the interactions between plants and microbes and minerals in the rhizosphere and how dynamic they are. And then maybe most importantly for this crowd, how important they are for carbon storage in soils and sort of our understanding of what constitutes soil health and how we can promote soil health. And you are the lucky ones that get to see a pretty much completely new talk that I haven't given in this form before, and I really just took this opportunity to pull together our rhizosphere work for this seminar. And normally I present things that are published and that are sort of easy to talk about. And this time I'm presenting a fair amount of unpublished work that I think, at least in its first draft form, came together pretty well and sort of highlights how dynamic these rhizosphere processes are and how important they are for us to think about in terms of carbon storage and soil health. And so maybe with that, I'll just start acknowledging my collaborators and students here. This work was performed under a couple of different grants through DOE and NSF, and I really just want to name tag Mariella, who's done most of the work that I'm presenting here. She's a GRFP and a student in my lab and has been for a few years now. And then my collaborator, Zoe Cardone at the Marine Biology Lab, who's really an expert at rhizosphere processes. Can you see my mouse pointer?
Yes.
Okay, great. So I'm interested in soil organic matter because it is the largest and most dynamic terrestrial carbon stock. It's more than twice the size of the biosphere and atmosphere pools combined. It really stores a lot of carbon. And I call it dynamic because it's continuously replenished through plant inputs, whether it's above ground leaves or woody material or below ground inputs like roots. And it is that carbon is released through biological processes, namely microbial processes that convert soil carbon back into greenhouse gases and release it into the atmosphere. So there's a dynamic exchange going on, but the overall soil carbon pool really resides within soil organic matter. And we care about soil organic matter for a number of reasons. Sorry, I'm having issues here advancing my slides. Oops. And we care about soil organic matter because of its implications for food security. We all want to grow crops in soils, and organic matter is a primary reason for soil fertility. Energy, biofuel biofuel crop production comes to mind. Climate, we can store carbon in soil organic matter and so can mitigate climate, and water, because organic matter is really important for the soil's function to filter out contaminants. And so really at the center of this talk are roots, because I think they're key to really addressing many of these various challenges that we are facing in terms of managing soil organic matter. And roots are so important because they're the main conduit of carbon into the soil, but because they are very active, they're alive, most of them, they also promote the microbial processes that release greenhouse gases into the atmosphere. And so really, when we think about how to manage soil organic matter, they're really at the center of what we need to think about. And so really the understanding that's emerging of how we can build up soil organic matter stock has at its core the somewhat recent realization that roots and associated microbes are really the main source of soil organic matter rather than above ground residues, whether it's leaves in forests or crop residues that are just sitting on the soil surface. Most of the organic matter that builds up over long periods of time is derived from root material and whatever is chewed on by microbes. And so you can think of that root material, that root cells that are coming off the individual roots, different metabolites and exudates that are being released by the roots to mobilize nutrients. They all collectively are often referred to as rhizodeposits, and they all feed microbes that live in the soil surrounding the root, what we often refer to as the rhizosphere. And those root materials, together with the microbial biomass that is established in the rhizosphere, then contribute to soil organic matter. And the key to stabilizing organic matter, and especially root-derived soil organic matter in soils, is the fact that these roots are in intimate contact with the soil, right? In contrast to, say, leaves that just get deposited on the surface of a soil, these roots grow right next to minerals. And these minerals often have really high surface areas. They're really reactive. They can bind not just nutrients, but also a lot of organic matter. And so a lot of that root material will inevitably bind to some minerals and then be sequestered. And that happens to a degree that is so strong that we now generally think that these mineral organic associations that form the root are quantitatively the most important carbon storage mechanism. And that is because once that organic matter is bound to a mineral, my student likes to scribble these little cartoons here, so you can think of these little red things here as a mineral, and then organic matter as this dark organic amorphous thing. But once these things form, these mineral organic associations, it's really hard for microbes to get the organic matter off the minerals again. And so the idea is that that organic matter then is locked up and can't be converted to greenhouse gases anymore. And that really is the emerging paradigm here, is that if we want to store more carbon in soils, what we should think about are strategies to get more root-derived organic matter into the ground, grow more roots, grow certain types of roots that deliver a lot of carbon into the ground, that maybe build up microbial biomass in some shape or form, and that then results in that newly formed organic matter being intimately associated with minerals in these mineral organic associations that protect organic matter from live microbes. And so there's a lot of talk about how we can manage soil sustainably, how we can adjust and adapt management strategies in order to funnel carbon into these mineral organic associations these days. The problem with that perspective, and I'm not saying it's not working, but one of the things that one should think about is that roots are not just conduits of carbon into the soil. So yes, there's a lot of carbon coming into the soil through roots, but there's also a lot of other things going on around roots that have to do with microbial activity, that have to do with water and nutrient uptake, and so forth. And roots, by being so active and interacting with the soil so much, really change not just the biology, not just the geochemistry, not just the hydrology, really all of those facets of that define the rhizosphere environment. So they really have a massive impact on what's going on in the soil around it that goes much beyond just being a funnel of carbon into the soil. And many of these processes have the potential to undermine mineral organic associations, to basically unlock the organic matter that is bound to minerals and make it accessible to microbes again. We've done some work early on looking at different types of compounds that are being released by roots, so-called exudates, and their ability to mobilize organic matter from minerals and make it more bioavailable for microbes. There's other changes ongoing in the rhizosphere that we know of, whether it's pH gradients or redox dynamics, water exchange, nutrient uptake, that can change the solubility of minerals, that can potentially dissolve minerals, that can potentially change sorption equilibria that would result in the mobilization of organic matter from mineral. But most of those processes are pretty poorly understood, and we don't really know the extent of these processes as well as the mechanisms. But there is certainly the potential to unlock organic matter from minerals, because roots tend to be pretty good at extracting things from minerals. They're designed to do that, to mobilize nutrients, so why wouldn't they be able to mobilize organic matter? And so that really is the driving question for the research that we've done in the rhizosphere, is how these processes influence not just the formation of mineral organic associations by adding carbon to the soil and bringing it close to minerals, but also the disruption of these mineral organic associations. And we are principally interested in answering the question of how this balance between these two processes impacts soil carbon storage over longer time scales, what the underlying mechanisms are, so how this all works, and then ultimately figure out a way how we can predict them better. And that is by incorporating some of these mechanisms into models that are able to predict some of these phenomena over longer time spans. So over the course of this talk, I just want to walk you through a couple of examples of how we address some of these questions here. And I'm going to start just with the first one, the long-term impacts of these rhizosphere dynamics of mineral organic associations. And for this, we took advantage of a soil chronosequence that shows really pronounced rhizosphere dynamics over time. And the soil chronosequence, for those of you who are not familiar with it, is basically a series of soils of different ages that formed under very similar conditions, whether it's climate or vegetation. And they allow you to basically have a snapshot in time of that soil developing. And so in this case, we have a series of 4, even though I'm just 5, and even though I'm just showing you 4 soils here that range in age from 50,000 years to 250,000 years, roughly, and that have really, really nice and unique rhizosphere zones develop in the subsoil. And those zones develop around these root channels, and these roots over time just really alter the mineralogy of the soil. And they do that, and which then results in this color change that you see really nicely down below. And those roots emerge after and get down to that soil depth in this at the second time point, and then expand these rhizosphere soils. And you see that really nicely in this data here that basically shows you that the extent of that rhizosphere soil once the root arrives at this time point here, and how it expands over time at the cost of what we call bulk soil here, which is the remaining soil. So the roots keep growing into these root channels and just expand this rhizosphere and weather away on the minerals and transform them over time. And we took these rhizosphere soils and bulk soils and subjected them to a number of different analyses to really get at how the mineral organic associations and ultimately carbon storage within these mineral organic association changes over time. And so one of the things my student Mariella did is to look at reactive minerals, and we're using an extraction to look at those. We call them short-range order phases here. And without going into further detail, it's pretty clear that in a rhizosphere soil, what you can see is that there's a pretty massive jump between the first and the second time point here in terms of the abundance of these reactive minerals. And that's exactly when those roots arrive. So something happens when those roots arrive that really stimulates the production of these reactive minerals. And then as these rhizospheres develop and roots keep growing into them, the abundance of these reactive minerals declines. And with that coinciding is the amount of carbon that is found in these rhizosphere soils. And this is a correlation that is pretty common in soils globally. The abundance of these reactive minerals and total carbon, which first informed our understanding of the importance of these mineral organic associations for carbon storage globally. And there's many meta-analyses now out there that show really nice 0.99 type correlations between the abundance of these short-range order phases and carbon stocks and soils. And so we see that in the rhizosphere too. But interestingly, there's this increase and then a decline as the roots arrive. When we look at the bulk soils here, what we see is that there's not a jump. It just sort of steadily increases over time with not much change. So quite different dynamics in a bulk soil that's not directly affected by the roots. So why would roots create these reactive minerals initially? And the answer is fairly easy. We know that roots are pretty involved in weathering primary minerals. So what they do in these deeper soils is they grow on rock, essentially, and they turn these primary minerals into these really reactive secondary minerals that are shown here as orange rust, essentially. They're iron oxides that are really reactive. They're tiny, so they have really high surface areas, but they also have very strong binding sites for organic matter. So what is happening here, seemingly, is that these roots and fungal hyphae grow on this rock, they weather away the rock, and the weathering products are these tiny little iron oxide minerals that then trap the organic matter that's brought in by these roots. So we wanted to look at this a little more closely in our chronosequence using a technique that's called Stickson scanning transmission X-ray microscopy. It's basically a high-resolution X-ray microscope that allows you to look at carbon and iron in our case. And you can see in those images here that are really at small scales, carbon in blue and iron in red. And again, we have our 4 time points. And I'm depicting here again sort of the amount of carbon that we found in these soils over time. And when you look at just these color maps, you see that there's these specks of red iron minerals, and they don't seem to be associated with a lot of carbon here. But as soon as we get to this time 2, there's lots of red specks again. In this case, they're actually looking purple, which indicates that there's red and blue together. And you mostly just see this diffuse, these diffuse patches of red and purple and blue, indicating this intimate association between iron and organic matter that forms here. And then over time, we see that less and less, and there's these red specks here, but not a lot of blue. So something happens over time where these minerals become more crystalline. They're aging. And when they become more crystalline, they have lower surface area and less ability to bind organic matter. So it seems, in our case, as if once that initial root weathering that root impact starts, we are transforming primary minerals to secondary minerals, and we sequester a lot of carbon in these mineral organic associations. But then over time, as the roots continue to exert their impact, these minerals age and they become less reactive and they sequester less carbon, and maybe the roots stimulate enough decomposition of that mineral associated carbon that the overall carbon stock declines. So there's a case to be made for root stimulated formation and then disruption of these mineral organic associations over this longer time spans. And you could maybe argue that in younger soils you would be more likely to be seeing formation of these mineral organic associations. In older soils, maybe their loss due to root impacts. Okay, so that was that chapter on sort of these long-term impacts that roots can have in soil carbon storage during pathogenesis, soil development. Now I want to talk a little bit about how we go about elucidating the underlying mechanisms. And one of the main challenges with that is that up to this point, it's been really hard to capture the dynamic microenvironments that emerge around roots. And roots are living creatures, and as I mentioned, they change pH, exudation, the water budget in the soil on very short timescales. And we think that in order to really get at some of the mechanisms that dictate how mineral organic associations form and how they are disrupted, we need to better understand these short-term dynamics. And I'm saying we, but I really mean my student Mariella, has done a really nice job over the last few years developing these rhizoboxes. And these rhizoboxes allow us to grow plants. In this case, small seedlings, but we can also grow them bigger, and continuously monitor biogeochemical parameters using these microsensors along individual roots that grow down these rhizoboxes. And we have a number of different microsensors that we install in this array. For example, microdialysis probes that allow us to passively capture pore water and measure dissolved organic carbon concentrations, different exudates or metals and nutrients. We have microbiosensors that I'll talk about in a minute that allow us to capture microbial activity on the same scale. And then we have different microelectrodes that allow us to measure things like oxygen, pH, redox potential, iron 2 concentrations that are all really good to constrain to what extent minerals might dissolve in a soil and maybe liberate organic matter. And so I'm showing you a couple of examples of data that we recorded along individual roots to demonstrate to you that there are different microenvironments that have potentially different effects on mineral organic associations. So this is data that we collected using these microdialysis probes. And what I'm showing you is a profile of DOC, which we use as a metric of the amount of root carbon that's being released from the roots. And so you see that over time, and you can imagine that the root is approaching and then arriving at the sensor. And then as soon as it arrives at the sensor, there's carbon shooting up, which we would expect because we know that root tips are really active. They're actively growing, they are exuding exudates actively and passively leaking a lot of carbon just from growing. And so this spike is not super surprising, but really interesting to detect. And then you see over time that there's less and less carbon being measured in the rhizosphere as these roots grow by the sensor. So we then can take that same sample and do mass spectrometry and to try to get at the different types of potentially exudates that are being released into the rhizosphere. And we do this with the Environmental Molecular Science Lab, EMSOL, at the Pacific National Lab. This is a DOE-funded institution, and we have user grants there to access their mass spec instrumentation and be able to work with their analytical chemists, Rosie and Renee in this case. And they've, for example, been helping us collect really nice targeted HPLC MS data on these samples, and that's what I'm showing you here, where we grouped a few different compounds that are the most abundant in these two plots here. You see some of these organic acids emerge pretty much right after the root arrives. And then there's this other group of compounds that, could be microbial metabolites that are much more abundant at later stages of root growth, so around mature roots. And so keep that in mind, that there are these potentially microbial metabolites emerging along older roots. Because when we look at the microbial activity around these roots, we see that microbial activity is ramped up along these older roots. It takes microbes a while to start becoming active, and we can measure that using that microbiosensor that I just mentioned. And in this case, this is a technique developed by Zoe Cardone at the Marine Biology Lab, the person I mentioned earlier that I collaborate with pretty closely, and these measurements were done with my student together. And it's a measurement that relies on fluorescent microbes that are genetically modified to fluoresce when they are active, that we can basically add to the soil. And then we visualize these fluorescent microbes when they become active using a fluorescent camera. And so what I'm showing you here on the left is a video of just the roots growing. They grow, centimeter or two per day. So you see that time lapse for a day. And then on the right, you see just the luminescence or fluorescence. And I just want to highlight this little point here where the root is actively growing, and you see sort of this hotspot emerge. And so this hotspot, every time it does that, is a flush of microbial growth. And you can just do this for a bunch of different roots and get image analysis done, and then plot this as luminescence, which basically equates microbial activity. And you see that at the very, very tip of the root, there's sort of this initial spurt of microbial activity, but then it takes a while for microbial activity to ramp up too. Which is consistent with a lot of data that we know that we have about microbial activity around roots. And it takes a while for roots to really colonize these new root regions. So again, we sort of see this by these two different zones emerge around roots, around fresh roots or young roots and older, more mature roots. And then finally, I want to just show you some of the biogeochemical data. And in this case, redox potential and pH. Again, low redox potential and low pH would indicate higher solubility of minerals and metals that we're interested in that potentially harbor organic matter. And so what we see here is that when the root arrives, so here's time 0, we count down, the root arrives, pH and redox potential tends to drop, meaning mineral solubility goes up. And then over time, as the root matures and maybe doesn't exude as much carbon, is not as actively growing, we see those values recover. And so you could argue that right around here is where you probably have the highest potential of mineral-organic associations to be disrupted because of these lower pH and redox potential values. And we haven't really gotten to the point where we can make where we can provide any definitive conclusions on that. But it's interesting to us that when we look at the iron, the dissolved iron around the root, it really correlates with DOC. And so it could mean that either there's lots of root carbon coming in and that's dissolving the iron, or it could mean that the iron is being dissolved by whatever root processes there are going on, and the DOC goes up because there's organic matter mobilized from minerals. So this is a question we can't answer yet, but I think it's interesting that we can measure that and we can start asking those questions in more detail. And so if I had to speculate a little bit about different microenvironments around roots and how they might impact mineral organic associations, um, I would probably broadly differentiate younger roots, the root tips that are really active in terms of releasing carbon, and then older stages that are maturing and differentiating. And we already know, based on literature, that rhizodeposition is probably highest around the younger roots, and microbial activity sort of starts increasing along these growing roots. And that is really also evident in our data, which is nice. And then when we look at our geochemical data, we see that these young root stages really promote a decrease in redox potential and pH, which increases solubility of minerals and therefore ion concentrations and dissolved ion concentrations, but also DOC concentrations. So maybe there's higher potential for disruption of these mineral organic associations formed by ion minerals. But the more mature root zones increase redox potential relative to the younger stages and pH. So maybe there's not as much mineral dissolution going on. We also don't see as much ion dissolution, which would corroborate that conclusion. And we see less DOC, but we see higher microbial activity. So maybe there's a case to be made that this is really where there's more potential for the formation of mineral organic associations, right? Your microbes are starting to bloom. They're probably turning over very fast. They're producing microbial biomass that can then bind to minerals. And so when you think about how roots might sequester carbon, that might also be an important consideration. Where along the roots those mineral organic associations are forming. Okay, so it's possible that along a single root you can have zones of for the formation and zones for the disruption of mineral organic associations. And we're doing work along these lines to really get at that, trying to pair these microscale measurements with more direct measurements of these mineral organic associations. So the final chapter that I want to just get at briefly here is how we go about incorporating these processes in predictive models and how we can then use them to inform our understanding of what these mineral organic associations do in the rhizosphere. And one of the main challenges has been that we can build these really complicated models that represent not just the above ground part, the plant, but also the below ground part, the rhizosphere, in quite a bit of detail. It's easy to put a lot of different reactions into these emerging models. We have the computational tools to do that. But especially when it comes to rhizosphere processes, we've had the challenge of not being able to really parameterize these models because it's so difficult to measure these different rhizosphere processes directly. And so what we were trying to do here is pair our Rhizobox approach, which is really good at measuring some of these biogeochemical parameters in the rhizosphere, with this really refined model called East-Dump Roots. And that model in particular is really good because Steve Yabuzaki, Neil and Fang at PNNL managed to integrate a plant biophysics model that was already established with a root model that incorporates root architecture and root growth dynamics with a reactive transport model that represents the biogeochemistry that we expect in the rhizosphere. So all the dynamics between, say, a mineral particle and organic matter and microbial activity and so forth. And we tried to integrate them in a way that would allow us to then predict the dynamics of mineral organic associations in the rhizosphere. And so I'm just showing you a couple slides to illustrate how we go about parameterizing this model. So we can take our rhizoboxes and just take photographs of the roots and then build a 3D model of our root network based on that. And then we can simulate rhizodeposition, a certain carbon allocation below ground that then is being released around the root tips. You see here carbon concentrations around the roots. So in this 3D model, basically the roots are growing everywhere, and then around the root tips there's carbon being released into the soil, the way we envision it. And then also simulate the influence that, for example, the roots have on water distribution, and couple that to transpiration. So rhizodeposition is directly coupled to photosynthesis and water distribution to transpiration. So there's really a mechanistic coupling between what's going on above ground and what's going on in the rhizosphere. And so to parameterize all of this, we took all these measurements in rhizoboxes. We subjected them to these day and night cycles, light off, light on, and then measured photosynthetic rate, transpiration rates, and provided numbers for the model. And then we also measured the below ground biogeochemistry. And so over these day and night cycles that we are showing here, so night is the white part, the orange yellowish part here is daytime, you see these diel or diurnal variations. Right? So you see carbon in the rhizosphere go up every single time you turn the light on, essentially, because all these photosynthates are being transported to the root and then are leaking out of the root, probably. And then during the daytime, when this carbon is coming in, you see also dips, sometimes more pronounced than others, in dissolved oxygen or redox potential. And what we think is happening here is that, just like before, when the root arrives and is pretty active, all this incoming carbon stimulates microbial oxygen consumption and reduces the redox potential. And so there are these really discernible diel variations in the rhizosphere biogeochemistry. And so we put these numbers in a model, and then we try to parameterize it, and then we try to validate it by comparing our simulations to the observations. And generally for DOC and pH dynamics and oxygen dynamics, we can generate a model output that matches that of these diurnal dynamics that we measured. Okay, but then more interestingly, we can try to predict the dynamics of mineral organic associations in the rhizosphere. And what we generally see based on the model outputs is that there are diel variations in the stability of in this case iron and organic matter associations. And what we see is that during the day, when lots of carbon is coming in and the oxygen is consumed and the redox potential goes down and we are dissolving these iron oxides, there's carbon released from these iron oxides. And then during the night, we actually reform these iron oxides because the rhizosphere becomes more oxidized again. And those iron oxides start binding carbon again. So we don't have data to confirm this model simulation, but it's an intriguing thought to think of these dynamics to be so dynamic that they, on the root surface at least, would cycle between the formation of mineral organic associations and the disruption of mineral organic associations over such short timescales. And it makes sense given what we know about plant root uptake. There are plant nutrient uptake by roots, where we have observed similar diel cycles where nutrients are mobilized during the day and they are taken up with the water uptake. And so it makes sense that the dissolution of these iron oxides that has as a consequence the mobilization of organic matter would behave similarly. And so again, ongoing work in my research group tries to tackle more broadly the dynamics of mineral organic associations in the rhizosphere. We are doing this sort of at the very mechanistic scale with batch experiments. We just had a paper out looking at how different exudates can stimulate the mobilization of organic matter from minerals. we're looking at it with real plant soil systems in a greenhouse and these rhizo boxes, and then we also have ongoing field experiments where we're looking at how root growth impacts labeled mineral organic associations that we had put out in the field at this point 4 years ago. So there's hopefully more to come if my students don't quit on me. But I'm going to leave you with a couple of take-home messages that I think are important for us to think about when we think about efforts to sequester carbon in soils and manage soils sustainably. And I hope I convinced you that rhizosphere processes are really important in regulating not just the formation of these mineral organic associations, but also the disruption, right? And that it really is the balance of those two processes that we need to consider when we think about root carbon and root activity and promoting that. And then secondly, we saw that these mineral organic associations can be pretty dynamic, both over short time spans and over long time spans, right? And that balance between formation and disruption may change depending on the scale and the ecosystem that we are considering. And then finally, that these mineral organic associations in the rhizosphere and the dynamics in the rhizosphere are really important just for general assessments of soil carbon storage potential. And so I just have a couple of thoughts in the end why we should care, essentially. And one question that's worth asking is if these mineral organic associations are really more dynamic than we previously thought, right? And this whole idea of just promoting the inputs of root-derived organic matter may not quite work. Like, how can we think about increasing carbon storage in mineral organic associations, right? So we have to think about soil type, right? Are there young soils that maybe have weatherable minerals? Can we add weatherable minerals, right? There's a huge push to add primary minerals and sequester carbon that way. Can we develop root traits that promote formation of mineral organic associations over disruption? I don't know. Maybe there's a way to do this, right? Can we think of agricultural practices that enhance formation rather than disruption, right? If you have really good crops that are good at nutrient mining, I would worry about the disruption part, right? The more plants are adept at mining for nutrients in soils, I think the better they will be at disrupting organic matter or disrupting mineral organic associations. We also know that mineral organic associations are rich harbors of other organic nutrients, nitrogen, phosphorus. Right? So if we recognize that these mineral organic associations, especially in the rhizosphere, are dynamic, how do we think about this nutrient pool? Right? And how can we think about it in terms of reconciling our demand for carbon storage, right? So getting more organic matter in these mineral organic associations, but also maybe taking advantage of these nutrient pools for crop production, right? It's a slow release fertilizer if it's dynamic and disrupted on a regular basis by root processes, right? Maybe we can harness that. And I know Dave McNair has been working on phosphorus in that direction. We have a project on organic nitrogen. So that's, I think, an interesting question going forward. Oops, sorry. And then finally, we know that climate change and land use change vegetation patterns, the diversity of above ground vegetation. There's stresses that are exerted on plants that will change their root activity and root dynamics. And how does that change the balance between formation and disruption of mineral organic associations? So I think those are all considerations that are really important for us to think about when we try to manage soil organic matter going forward. So that's it for me. Thank you for your attention. This is just my group, and I'm happy to answer questions if you have any.
Thank you so much. Are there any questions?
Marco, Chris Matoka here. Nice job. And in a complex system, I have a question. on the chronosequence site, were there differences in mineralogy over time, and how that might impact the carbon storage?
Yeah, so we did XRD and Mossbauer, XRD mostly to look at the clay minerals, and then Mossbauer to speciate the iron oxide minerals a little better. And it really was driven by the short-range order minerals, the carbon dynamics. So there are obviously other clay minerals in there. There's lots of kaolinite that changes over time. There's other ferrosilicates, but it really was the short-range order mineral, the extractable pathogenic oxides, if you want, that were the most correlated with the carbon dynamics in the system. And among that, just to be a little more specific, the Mossbauer results really showed nicely a correlation between a really disordered goethite, nanogoethite mineral, and carbon storage. So it's not just just broadly polycrystalline materials. It seems to be this nanogoethite phase that forms in these particular soils.
Marco, Dave McNear, how are you? I hope you can hear me. I'm in a bit of a chaotic environment here.
I can hear you very well. Yeah, you're a little...
Okay.
Yeah.
It's good to see you. Though I'll ask a quick question. I just wanted to know if you could comment about the quality of the inputs. And maybe you already said this because I came in a little bit late, but you talk a lot about mineral associated, which tend to be a certain, like exudates and the low molecular weight stuff, but roots are more than that. And when we talk about carbon stabilization and sequestration, what do you think about root quality?
Yeah, I think that's a good point. And obviously these probes that we have to measure root carbon inputs are really only able to capture these metabolites and soluble inputs that we measure. And I would speculate that most of them are of microbial nature anyway. But to your broader point, I think that in terms of thinking about carbon sequestration and association of organic matter with minerals, that the root composition will have an effect. I'm still wondering to what extent it will supersede the effect that microbial communities have. That, as I mentioned, sort of the thinking now becomes more that most of it goes through the microbial mill anyway, and that what is left behind is mostly microbially processed material, so maybe it doesn't matter all that much what the composition of the root is. I think that's still a question to be really nailed down, and it probably will depend on the systems and how much bioturbation there really is. How's that for a non-answer, Dave?
I'll take it. I'll take it.
Marco, this is Rebecca McCulley. I enjoyed your talk. It identifies some very complex spatial and temporal patterns that kind of get a little bit mind-boggling. But I think some about hydraulic redistribution, and in your modeling exercise, where you're talking about the day-night dynamics that you get with it. It sounds like you think nighttime being a possible reformation of the carbon mineral complexes in daytime as oxygen is consumed and destabilizing. Have you thought at all about how plants that might be doing the opposite sort of scenario with their water, how that would be impact any of that?
Yeah, we've thought a fair amount about it, and mostly driven by my collaborator Zoe Cardone, who's done quite a bit of work on hydraulic redistribution. And In our rhizoboxes, I think they're just so that they're fairly wet, so there's not a lot of additional water coming in at night or anything. But it's been a challenge for us to really think about it in the context of the rhizoboxes, because we don't really have the same way of visualizing water content at the same scale. And we rely on these bulk measurements of water content in the rhizosphere, in the rhizoboxes, and that's insufficient because that almost doesn't change through day and night cycles. And that's probably one of the main shortcomings in the model too, is that we have to rely on best guesses in terms of how the distribution in the rhizosphere itself actually changes. Based on transpiration rates, we are just sort of scaling it based on transpiration rates. So we know how much water is being transpired, how much water must be taken up by the root, and then have a simple diffusion model in the water, in the soil to sort of account for that. But it would be better to have direct measurements. But yeah, to get at sort of the more conceptual point, I think there's certainly cases where you could expect wetter soils around the root at night when transpiration subsides, and then maybe you have root respiration consuming oxygen, and more reducing conditions during nighttime and microbial activity during nighttime. That is also something that I think you could envision in a given system.
Thank you.
Hello, I have a question. Thank you. Great talk. I enjoyed it. So in your modeling approach, you use an hourly time step in the Rhizosphere biochemical model, but this will be more frequent than time step than in many crop simulation models. I was wondering, do you think that this hourly time step and considering the diurnal cycling you described will be key when coupling an improved organic matter model with a crop model, or that there is an opportunity to learn from your research and describe this process in a more simplified way before this coupling, or how do you see this happening?
Sorry, I had trouble understanding your question.
Yes, so
were you asking about whether it's possible to scale the model, or?
Yes, so the Rhizosphere biochemistry model that you described works on an hourly time step, from what I see,
right.
I think.
Yeah.
And so many crop models work on a daily time step, or maybe they consider hourly time step just for some of the processes. So when coupling this with a crop simulation model, maybe to improve organic matter dynamics, will it be key to keep this hourly time step, or there is a possibility to simplify before scaling?
Yeah, that's a very good and important question. It is possible to run the model at longer timescales, and there's many examples of how these models have been run for years and decades. Then it becomes computationally maybe a little more challenging, and you wouldn't probably do this on a single plant basis where you have every a voxel be the size of a micron or something like that. But we've started to do modeling at our field site, where we are looking at essentially the same processes just seasonally, and are trying to simulate springtime plant productivity increases, and grass is basically growing for a couple of months and developing the root networks and then dying. So you could do similar things with a crop model type simulation, right? In that case, you're probably going to scale down the level of resolution, and you're not going to be able to resolve your root tip and your mature roots at the same level, or maybe even just individual roots the way we are doing, we basically have an exponential function that replaces the root network that just shows the depth profile of root distribution in that case, and have simplified the model there. But in the landscape, that's, I think, as good as we can get at this point to reflect the differences in root depth distribution across the landscape.
I was curious about a little bit more detail about the nature of mineral organic matter associations. Like, would you say, and I'm sure it depends on the mineralogy, but like, is it fair to think about it as organic matter bound to the surface of minerals, or more of a integration, like they co-precipitate and they're kind of intertwined? Or sometimes I hear about adsorption, and adsorption doesn't seem like a very strong binding. So I've been like confused about that.
No, adsorption can be very strong, but it's a good question. And when you read the more, maybe applied literature that more broadly characterizes mineral associated organic matter, they lump a lot of different things into that term, right? And it will really depend on the soil what will be the predominant type of mineral organic associations in a given soil in that fraction, operational fraction. And it's interesting because I used to think that in soils that are mostly under agricultural use, so more soils that we think of as having a fair amount of 2-to-1 clays, where we attribute carbon storage mostly to those. You also see in recent years more and more papers coming out that actually show that now when you extract iron, you have much better correlations between the pathogenic oxides, even in those soils, than with some of the fill silicates. And so I think in a lot of cases, you ultimately have some form of microaggregate where these pathogenic oxides are really important and cement together fill silicates and organic matter and pathogenic oxides. And what I'm starting to come to believe a little more is that the fill silicates are important for carbon storage, but maybe not because of their surface area, but because they create certain microbial habitats that are really beneficial to necromass being stabilized. So it's not necessarily that they forms, direct absorptive bonds with the organic matter, but that their physical properties have something that really lends themselves to being a good microhabitat for microbes in the first place. Because when you look at the clay fraction and you look at fill silicates, you always have huge biomasses in them too, right? So I think in the case of fill silicates, it's still more that they sort of nurture microbes in a way that produces more necromass. And in the case of pathogenic oxides, I believe that they are more important in terms of actually chemically sequestering it.
Cool, that helps a lot. And you would say that the iron oxides, even in soils that are not like altisols or oxisols, they could still be important in moderately weathered soils as well.
Yeah, it's just one of those things where historically, because it's not something that you measure in agricultural soils a ton, right? Because it wasn't part of the soil classification scheme. In andesol, yes, you measure oxalate extractable iron and aluminum because it's part of the scheme, right? But why would you do that in some moss or some are random? But when you do, you do find pretty strong correlations. Which is not to say fill silicates don't show correlations, but with carbon stocks, but I think that they've been underappreciated in that context. Yeah.
Cool, thanks.
Yeah.
Any other last questions? Getting close to 4. All right. Well, thank you so much. That was a really good talk. So really appreciate you giving our seminar today.
Yeah, thanks, and I hope that at some point I'll be able to visit in person without weird restrictions and mask mandates and so forth. It'd be fun.
Cool. Well, have a
All
good
right.
weekend.
Thank you. You too. Bye-bye.
You.